This is audio recording for visually impaired students. Drugs are chemicals of low molecular mass. These interact with macromolecular targets and produce a biological response. When the biological response is therapeutic and useful, these chemicals are called medicines and are used in diagnosis, prevention, and treatment of disease. Most of the drugs used as medicines are potential poisons. If taken in doses higher than those recommended, use of chemicals for therapeutic effect is called chemotherapy. Drugs can be classified mainly on criteria outlined as follows. A. On the basis of pharmacological effect. This classification is based on pharmacological effect of the drugs. It is useful for doctors because it provides them the whole range of drugs available for the treatment of a particular type of problem. For example, analgesics have pain-killing effect, antiseptics kill or arrest the growth of microorganisms. On the basis of drug action. It is based on the action of a drug on a particular biochemical process. For example, all antihistamines inhibit the action of the compound histamine, which causes inflammation in the body. There are various ways in which action of histamines can be blocked. You will learn about this in section 16.3.2. C. On the basis of chemical structure. It is based on the chemical structure of the drug. Drugs classified in this way share common structure features and often have similar pharmacological activity. For example, sulfonamides have common structural features given below. D. On the basis of molecular targets. Drugs usually interact with biomolecules such as carbohydrates, lipids, proteins, and nucleic acids. These are called target molecules or drug targets. Drug possessing some common structural features may have the same mechanism of action on targets. The classification based on molecular targets is the most useful classification for medicinal chemists. Macromolecules of biological origin perform various functions in the body. For example, proteins which perform the role of biological catalyst in the body are called coenzymes. Those which are crucial to the communication system in the body are called receptors. Carrier proteins carry polar molecules across the cell membrane. Nucleic acids have coded genetic information for the cell. Lipids and carbohydrates are structural parts of the cell membrane. We shall explain the drug target interaction with the example of enzymes and receptors. A. Catalytic action of enzymes. For understanding the interaction between a drug and an enzyme, it is important to know how do enzymes catalyze the reaction. In their catalytic activity, enzymes perform two major functions. The first function of an enzyme is to hold the substance for a chemical reaction. Active sites of enzyme hold the structured molecule in a suitable position so that it can be attacked by the reagent effectively. Substrates bind to the active sites of the enzyme through a variety of interactions such as ionic bonding, hydrogen bonding, van der Waals interactions, or dipole-dipole interactions. 2. The second function of an enzyme is to provide functional groups that will attack the substrate and carry out chemical reaction. B. Drug-enzyme interactions. Drug inhibit any of the above-mentioned activities of enzymes. These can block the binding site of enzymes and prevent the binding of substrate or can inhibit the catalytic activity of the enzyme. Such drugs are called enzyme inhibitors. Drugs inhibit the attachment of substrate on active site of enzymes in two different ways. Drugs complete the natural substrate for their attachment on the active sites of enzyme. Such drugs are called competitive inhibitors. 2. Some drugs do not bind to the enzyme's active sites. These bind to a different site of enzymes, which is allosteric site. This binding of inhibitor as allosteric site changes the shape of the active site in such a way that substrate cannot recognize it. If the bond formed between an enzyme and an inhibitor is a strong covalent bond and cannot be broken easily, then the enzyme is blocked permanently. The body then degrades the enzyme inhibitor complex and synthesizes the new enzyme. Receptors are proteins that are crucial to body's communication process. Majority of these are embedded in cell membrane. Receptor proteins are embedded in the cell membrane in such a way that their small part processing active site projects out of the surface of the membrane and opens on the outside region of the cell membrane. In the body, message between two neurons and that between neurons to muscles is communicated through certain chemicals. These chemicals, known as chemical messengers, are received at the binding site of receptor proteins to accommodate a messenger shape of the receptor site changes. This brings about the transfer of message into the cell. Thus, chemical messenger give message to the cell without entering in the cell. There are a large number of different receptors in the body that interact with different chemical messengers. These receptors show selectively for one chemical messenger over the other because their binding sites have different shape, structure, and amino acid composition. Drugs that bind to the receptor sites and inhibit its natural functions are called antagonists. These are useful when blocking of messages is required. There are other type of drugs that mimic the natural messenger by switching on the receptor. These are called agonists. These are useful when there is a lack of natural chemical messenger. Overproduction of acid in the stomach causes irritation and pain. In severe cases, ulcers are developed in the stomach. Until 1970, only treatment for acidity was administration of antacids such as sodium hydrogen carbonate or a mixture of aluminium and magnesium hydroxide. However, excessive hydrogen carbonate can make the stomach alkaline and trigger the production of ever more acid. Metal hydroxide are better alternatives because of being insoluble. These do not increase the pH above neutrally. These treatments control only symptoms and not the cause. Therefore, with these metal salts, the patients cannot be treated easily. In advanced stages, ulcers became life-threatening and its only treatment is removal of the affected part of the stomach. A major breakthrough in the treatment of hyperacidity came through the discovery according to which a chemical histamine stimulates the secretion of pepsin and hydrochloric acid in the stomach. The drug cimetidine was designed to prevent the interaction of histamine with the receptor present in the stomach wall. This resulted in release of lesser amount of acid. The importance of the drug was so much that it remained the largest selling drug in the world until another drug, nantidine, was discovered. Histamine is a potent vasodilator. It has various functions. It contracts the smooth muscles in the bronchi and gut and relaxes other muscles, such as those in the walls of fine blood vessels. Histamine is also responsible for nasal congestion associated with common cold and allergic response to pollen. Synthetic drugs, bromophenramine and terfenadine, act as antihistamines. They interfere with the natural action of histamine by competing with histamine for binding site of receptor where histamine exerts its effect. Now the question above that arises is, why do above mentioned antihistamines not affect the secretion of acid in stomach? The reason is that anti-allergic and antacid drugs work on different receptors. Tranquilizers and analgesics are neurologically active drugs. These affect the message transfer mechanism from nerve to receptor. Tranquilizers are a class of chemical compounds used for the treatment of stress and mild or even severe mental disease. These relieve anxiety and stress, irritability, and or excitement by inducing a sense of well-being. They form an essential component of sleeping pills. There are various types of tranquilizers. They function by different mechanisms. For example, noradrenaline is one of the neurotransmitters that plays a role in mood changes. If the level of noradrenaline is low for some reason, then the signal sending activity becomes low and the person suffers from depression. In such situations, antidepressant drugs are required. These drugs inhibit the enzymes which catalyze the degradation of noradrenaline. If the enzyme is inhibited, the important neurotransmitter is slowly metabolized and can activate its receptor for a long period of time, thus counterating the effect of depression. Iproniazid and phenazines are two such drugs. Some tranquilizers, namely chlorodial oxide and meprobamate, are relatively mild tranquilizers suitable for relieving tension. Equantil is used in controlling depression or hypertension. Derivatives of barbituric acid, example amytal, nambutal, luminal, and sacchin, constitute an important class of tranquilizers. These derivatives are called barbiturates. Barbiturates are hypnotic, that is, sleep-producing agents. Some other substances used as tranquilizers are Valium and serotonin. Analgesics reduce or abolish pain without causing impairment of unconsciousness and mental confusion, incoordination or paralysis or some other disturbance of nervous system. These are classified as follows: non-narcotic analgesics, narcotic drugs. Non-narcotic analgesics, aspirin and paracetamol belongs to the class of non-narcotic analgesics. Aspirin is the most familiar example. Aspirin inhibits the synthesis of chemicals known as prostaglandins, which stimulate inflammation in the tissues and cause pain. These drugs are effective in relieving skeletal pain, such as due to arthritis. These drugs have many other effects, such as reducing fever and preventing platelet coagulation. Because of its anti-blood clotting action, aspirin finds use in prevention of heart attacks. 2. Narcotic analgesics. Morphine and many of its homologues, when administered in medicinal doses, relieve pain and produce sleep. In poisonous doses, these produce stupor, coma, convulsions, and ultimately death. Morphine narcotics are sometimes referred to as opiates since they are obtained from the opium poppy. These analgesics are chiefly used for the relief of postoperative pain, cardiac pain, and pain of terminal cancer and childbirth. Disease in human beings and animals may be caused by a variety of microorganisms such as bacteria, virus, fungi, and other pathogens. An antimicrobial tends to destroy and prevent development or inhibit the pathogenic action of microbes such as bacteria, virus, or other parasites selectively. Antibiotics, antiseptics, disinfectants are antimicrobial drugs. Antibiotics. Antibiotics are used as drugs to treat infections because of their low toxicity for humans and animals. Initially, antibiotics were classified as chemical substances produced by microorganisms that inhibit the growth or even destroy microorganisms. The development of synthetic methods has helped in synthesizing some of the compounds that were originally discovered as products of microorganisms. Also, some purely synthetic compounds have antibacterial activity, and therefore definition of antibiotic has been modified. An antibiotic now refers to a substance produced wholly or partially by chemical synthesis, which is low concentration, inhibits the growth or destroy microorganisms by intervening in their metabolic processes. The search for chemicals that would adversely affect invading bacteria but not the host began in the 19th century. Paul Ehrlich, a German bacteriologist, conceived this idea. He investigated arsenic-based structure in order to produce less toxic substances for the treatment of syphilis. He developed the medicine known as salvarsan. Paul Ehrlich got Nobel Prize for Medicine in 1908 for this discovery. It was the first effective treatment discovered for syphilis. Although salvarsan is toxic to human beings, its effect on bacteria and spirochete, which cause syphilis, is much greater than on human beings. At the same time, Ehrlich was working on azodize also. He noted that there is similarity in the structure of salvarsan and azodize. The AS linkage present in arsenophenamine resembles the NL linkage present in azodize in the same sense that arsenic atom is present in place of nitrogen. He also noted tissues getting colored by dyes selectively. Therefore, Ehrlich began to search for the compounds which resemble in structure to azodize and selectively bind to bacteria. In 1932, he succeeded in preparing the first effective antibacterial agent, pronotosil, which resembles in structure to the compound salvarsan. Soon it was discovered that in the body pronotosil is converted to a compound called sulfonylamide, which is the real active compound. Thus, the sulfur drugs were discovered. A large range of sulfonamide analogues was synthesized. One of the most effective is sulfapyridine. Despite the success of sulfonamides, the real revolution of antibacterial therapy began with the discovery of Alexander Fleming in 1929 of the antimicrobacterial properties of Penicillium fungus. Isolation and purification of active compounds to accumulate sufficient material for clinical trials took 13 years. Antibiotics have either sedal effect or static effect on microbes. A few examples of the two types of antibiotics are as follows: bactericidal penicillin ofloxacin aminoglycosides, bacteriostatic tetracycline, chlorophenicol, erythromycin. The range of bacteria or other organisms that are affected by a certain antibiotics is expressed as a spectrum of action. Antibiotics which kill or inhibit a wide range of gram-positive or gram-negative bacteria are said to broad-spectrum antibiotics. Those effective mainly against gram-positive or gram-negative bacteria. They are narrow-spectrum antibiotics. It affects against a single organism or disease. They are referred to as limited-spectrum antibiotics. Penicillin G has a narrow-spectrum. Ampicillin or amoxicillin are synthesized modification of penicillins. These have broad spectrum. It is absolutely essential to test the patients for sensitivity to penicillin before it is administered in India. Penicillin is manufactured at the Hindustan Antibiotics in Pimpton and in private sector industry. Chloramphenicol, isolated in 1947, is a broad spectrum antibiotic. It is rapidly absorbed from the gastrointestinal tract and hence can be given orally in case of typhoid, acute fever, certain form of urinary infections, meningitis and pneumonia. Vancomycin and ofloxacin are the other important broad-spectrum antibiotics. The antibiotics diacidazoline is supposed to be toxic towards certain of cancer cells. B. Antiseptics and disinfectants. Antiseptics and disinfectants are also the chemicals which either kill or prevent the growth of microorganisms. Antiseptics are applied to the living tissues such as wounds, cuts, ulcers and diseases, skin surfaces. Examples are furosemide, soframycin, etc. These are not ingested like antibiotics. Commonly used antiseptic Dettol is a mixture of chloroxylenol and terpenol. Berthianol is added to soaps, impart antiseptic properties. Iodine is a powerful antiseptic. Is 2 to 3% solution in alcohol. Water mixture is known as tincture of iodine. It is applied on wounds. Iodoform is also used as an antiseptic for wounds. Boric acid is dilute aqueous solution is weak antiseptics for you. Disinfectants are applied to inanimate objects such as floor, drainage system, instrument, etc. Same substances can act as an antiseptic as well as disinfectant by varying the concentration. For example, 0.2% solution of phenol is antiseptic while its 1% solution is disinfectant. Chlorine is the concentration of 0.2 to 0.4 ppm in aqueous solution and sulfur dioxide is very low concentration disinfectants. Antibiotic revolution has provided long and healthy lives to people. The life expectancy has almost doubled. The increased population has caused many social problems in terms of food resources, environmental issues, employment. To control these problems, population is required to be controlled. This has led to the concept of family planning. Anti-fertility drugs are of use in this direction. Birth control pills essentially contain a mixture of synthetic estrogen and progesterone derivative. Both of these compounds are hormones. It is known that progesterone suppresses ovulation. Synthesis progesterone derivatives are more potent than progesterone. Norethindrone is an example of synthetic progesterone derivative most widely used as anti-fertility drug. The estrogen derivative which is used in combination with progesterone derivative is ethinyl estradiol. Thanks for watching.